Okay, so hello once again. So this time, this would be prelim lecture 4 covering bacterial genetics. Okay, so the learning outcomes for this topic are the following. So at the end of the discussion, you are expected to be able to describe the characteristics of a bacterial genome, to identify mechanisms of gene transfer that will occur in bacteria, and to recognize the clinical importance of the ability of bacteria to transfer genes to other bacteria with and between species. Okay, so of course, when we're talking about genetics, the chromosome or the bacterial genome will be at the core of it, of course. So the chromosome, the bacterial chromosome or the bacterial genome is a single long piece of circular double-stranded DNA that would contain about 2,000 to 4,000 different genes, genes that may code for, of course, their morphology, their uh, characteristics. And of course, we have the plasmids. Okay, so apart from the genetic material, they do have very small circles of DNA that can replicate independently and may code for abilities that they may not have been born with. Okay, so this would include toxins, okay, ability to resist antibiotics, or ability to produce sex pilus. Okay, so some bacteria may acquire this. Some may not really be born with it, but they can collect it from other organisms. Okay, so by talking about collecting genes from other organisms, we refer to it as genetic transfer. Okay, so genetic transfer may occur vertically, such as what happens during binary fission. So from mother cell to offspring. So when the mother cell divides into two daughter cells, so the genes are transferred from the mother to the offspring. However, genes can be transferred not from the mother to the offspring, but from one bacterial cell to another bacterial cell that's completely unrelated. That's what we call the horizontal gene transfer. Now, horizontal gene transfer can be in the form of conjugation, transformation, or transduction. So we will go through them one by one. So let's start with conjugation. So for this, we have the F plus conjugation. Okay, so this is bacterial mating. Okay, bacterial copulation. Okay, this usually occurs in enteric or gram-negative bacteria. Okay, so in this flash photo, okay, so you'll see, let's allow to restart first. Okay, so you'll see here one, the yellow one, an F plus, which is a male. Okay, it has a plasmid that codes for the production of sex pilus. Okay, it can serve as a genetic donor. So the sex pilus adheres to an F negative, yung orange, it's a female. Okay, so one strand of the plasmid is transferred onto the female. Okay, and then they are now both able to copy it. So they have two copies of the strand, and then now the female, the one that's initially a female, has become an F positive or a male bacteria or bacterium, and it is now able to code for the production of a sex pilus. Okay, so the yellow one is a male, F positive, which means it can produce sex pilus, and it has donated its plasmid that codes for that particular ability onto an F negative, or a female. Okay. So once a strand of the plasmid is transferred, it is automatically copied, allowing both to become F positive, and they are now both able to produce the sex pilus. Okay, so recall sex pilus, which has been discussed in structures also. Okay, so this is very important in conjugation. Okay, so that's F plus conjugation. Now, here we have resistance plasmid conjugation. Technically the same. It's just that the plasmid that's being shared is an R plasmid, a plasmid that can help bacteria resist some forms of antibiotic. So here, the yellow one is still the F plus and the R positive donor, okay, donating the R plasmid onto an F negative, R negative. Okay. So now the recipient is an F positive, R positive bacterium. Okay. So R positive plasmid is donated onto an R negative, F negative recipient. Once it's shared, it is automatically replicated to produce 2 strands. And so now the recipient who was previously R negative or non-resistant and F negative, no, unable to produce sex pilus, can now produce sex pilus. Okay? So that's how they share, no, properties. Then we have transformation. Okay, so transformation occurs after the uptake of DNA fragments from the environment. So when a bacterium dies, okay, it breaks up into pieces, and so some of its genetic material may be free swimming in the environment. No? So this transformation as a process of gene transfer may occur between 2 different species. But what's important is that this process of gene transfer will occur only to a competent bacteria. Now, what we mean by that? So a competent bacteria are able to accept naked DNA from the environment. So how can they become competent? Okay? Competent bacteria are those that possess the REC-A protein. Okay, so it's a critical enzyme in the process of transformation. It catalyzes the pairing of the single-stranded DNA with the complementary regions of the double-stranded DNA. Okay. So this is a competent bacterium with their REC-A protein. So the free DNA enters the competent bacterium. And because of the REC-A protein, the accepted donor DNA is able to attach to the host DNA, transforming the host bacterium into a new version. So that's transformation, simply absorbing genetic material from the environment causes the bacteria to transform into something that's new. Okay. And then we have the transduction. Okay, so transduction means to carry across. Okay, may tighatid. Okay, so sinong tighatid? Okay, so transduction is the transfer of genetic material or DNA fragments from one bacterium to another by a or through a bacteriophage. So bacteriophages are viruses. Okay, now there are 2 types of bacteriophages that initiate transduction. So we have here lytic or virulent phages, the T phages, and we also have the temperate phage or the phage lambda. Okay, now viruses like bacteriophages undergo reproduction. No? So or they don't we don't really use the term reproduction, but no, they replicate rather. Okay, so they replicate in 2 ways. The lytic or the virulent phage usually replicates through the lytic cycle. Okay, so it involves the production of viruses using a host cell to manufacture more viruses, and then the viruses would burst out of the host bacterium. That's why we call it virulent phage. It will cause its host bacteria to die. okay, to lyse, okay, so that the new virions can come out of the host cell. Now, the temperate phage or the phage lambda undergoes replication through the lysogenic cycle. Okay, so this type of replication involves the incorporation of the viral genome into the host cell genome, creating the so-called prophage. So it does not kill the host cell agad-agad, no? So hinay-hinay, it would kill the host slowly but surely. It would infect the host first, no? So the temperate phage would release its genetic material and cause it to incorporate or mix with the host cell's genome. Okay, so once they mix, we call that the prophage. Okay, so when it creates its smaller versions, no, it will create virions. The virions will now contain the prophage, no, so it has both the genetic material of the prophage, the temperate phage, and the bacterial host. No? So it's a mixture of genes. So that's what happens with temperate phages. Okay, so lytic or virulent phages will simply create virions by using the machinery of the bacterial host, whereas temperate phages usually incorporate their own genetic material with the bacterial genetic material, producing the prophage. No? So that's how different they are. Okay. So there are actually two forms of transduction. We have the generalized transduction and the specialized transduction. Now, in generalized transduction, when the lytic phages replicate, the capsid sometimes assembles around a small fragment of the bacterial DNA, like this one, right? So they assembled around a genetic material that's not viral by nature, but rather bacterial. Okay? So it appears to be a virus, a bacteriophage, but it carries a bacterial DNA. Okay? So it causes the transfer of one of the genetic material of another bacteria onto another bacterium. Okay? So that's generalized transduction. Okay, so let's watch again. So that's the viral genome, and they undergo replication and maturation of the proteins, and then assembly. So during the assembly, a genetic material is encased in a capsid. And so it infects another bacteria, but the genetic material it releases is actually that of the first bacterial host. And so now this bacteria has gained genetic material from another bacterial host. Okay, so that's generalized transduction. Now let's look at specialized transduction. Okay, so remember, specialized transduction is initiated by the temperate phage, which undergoes the lysogenic cycle as it replicates. Hence, the bacterial organism that falls prey to this type of gene transfer undergoes lysogenic conversion. What happens? During lysogenic conversion, the phage is allowed to take a bit of the adjacent bacterial cell. Okay, so nagmimix, there's a prophage formation. So when it transfers genetic material onto another bacterial host, it does not just carry viral DNA with it, but it also carries the bacterial DNA with it. So it's a combination. Okay. So cells now, bacterial cells now exhibit new properties. So those who were previously non-pathogenic may become pathogenic now after undergoing lysogenic conversion. Okay. Cells are also immune to reinfection by same phage. That's what they acquire after bacterial cells undergo lysogeny conversion. Now let's take a look. So here comes a temperate phage with the viral genome. Okay, it infects the bacterium with its viral genome. So the viral genome is allowed to combine with the host bacteria, creating the prophage. Okay. So once they replicate genetic materials, uh, the genetic material may carry a portion of the host and the temperate phage. So it's a combination. So see, all its offsprings, the new virions, carry a combination of the DNA of the bacteria and that of the virus. And so when it infects another bacteria, the bacteria now has access to both phage DNA and bacterial DNA. Ayan. Kaya naman yung dati ay hindi matapang, ngayon ay nagiging matapang na bacteria. Sino-sino ba sila? Examples of organisms, bacterial organisms that are usually prey to lysogeny conversion are Corynebacterium diphtheriae, Streptococcus pyogenes, Clostridium botulinum, and Vibrio cholerae. Ayan. So again, in specialized transduction, there's prophage formation. Okay, wala yan in generalized transduction. Okay, now genetic variation also occurs. Of course, there would always be mutation, especially when bacterial cells are exposed to, say, radiation. No? So mutation occurs when there's any change in the DNA-base sequence. So this may occur during exposure to radiation. Okay, and then of course, okay, because of the presence of transposons, so transposons are also known as jumping genes. No, these are mobile genetic elements that may cause plasmids of a different type to combine with another plasmid, or plasmids to combine with the genetic material. Okay, so transposons may cause plasmid to integrate into the main chromosome. So this is also responsible for spread of antibiotic resistance genes. Okay. So that ends our very short discussion on bacterial genetics. No, we will not be going too deep into this because we still have a lot to discuss and cover for part of the introductory topics for prelims. Okay, so hanggang dun lang tayo muna for genetics. Okay, so these are your references. Of course, again, you may use any other reference books that you have. You can use the Lost or Jowett's or Murray, no, if you have them. Thank you for listening.